Welcome to another episode of Drops of Science, the multilingual podcast that brings you the latest updates and discoveries in natural sciences. Our first story today takes us to China, specifically to Jiuzhaigou, a UNESCO World Natural Heritage Site famous for the stunning beauty of its wetlands, largely formed on tufa deposits. Here, trees and shrubs display a unique feature: dense and abundant roots with an intense red coloration during the summer. However, the mechanism behind this phenomenon have long remained unexplored. Researchers from Sichuan University in China have decided to shed light on this, mostly because the wetlands are threatened by degradation due to natural and human activities. They therefore positioned transects inside the Jiuzhaigou Mountain Park and recorded the plant species present, the percentage of plants with root tips, or whisker-like structures that emerge from some plants and develop in water. They measured the same tips, and through monthly photographs, they obtained the seasonal variation in the color of the root tips. Part of the roots was then collected and taken to the laboratory to be sectioned and to study their internal structure, cortex, parenchyma, and stele. Finally, environmental and physical data on water were collected. The results of the study showed that apical tips arise at the base of the stem and float in the water, showing a clear growth area, white or pink tip, a bright submerged part, red or orange, and a darker exposed part. The length varies from species to species, with some salix exceeding 80 cm. Anatomically, they show a lot of parenchyma, useful for gas exchange, and little xylem, tissue useful for water transport, which suggests an adaptation to the submerged environment. The color changes are seasonal. In summer, autumn, the root tips are red, pink or orange, while in winter-spring they tend to brown or grey. Chromatic variations were strongly correlated with air temperature, solar radiation, precipitation, and nitrate concentration. In particular, the color became more intense in basic pH conditions, while in acidic conditions it turns yellow. Light and temperature play a fundamental role. In fact, they promote the oxidation of pigments, intensifying the coloration. The main pigments found are proanthocyanidins, known for their response to light. All these elements indicate that apical roots can act as real environmental bioindicators. They are easily accessible structures, cheap to monitor and potentially scalable on large scale. Their chromatic response is dynamic, rapid, visible, and synchronized with environmental parameters, and their color variation could reflect microclimatic changes, thermal stress, or exposure to UV rays. Let's stay on the topic of coloration, but shift our focus to animals to talk about melanism in viperids. Coloration in animals is often an indicator of important information on the state of health, the reproductive phase, or interactions with conspecifics. In reptiles, however, coloration also plays a crucial role in thermoregulation. Darker shades absorb heat better, promoting faster body heating. Melanism, or an excess of dark pigmentation, is included among the color variants. It is a well-documented phenomenon in various reptile species, but its evolutionary origins and ecological relevance remain unclear. A study made at the University of Pavia examines the presence of melanism in viperids on a global scale, investigating its relationship with climatic variables such as temperature and precipitation in the context of the Bogart rule, which associates dark coloration with a thermal advantage in cold environments. The study also included a phylogenetic analysis to evaluate the ancestral state of the trait and its evolutionary stability. The data comes from iNaturalist, an open-access citizen science platform that collects naturalistic observations from around the world. The researchers analyzed a report of 139 species of viperids, recording cases of hypermelanism that happen when individuals are completely black on the back. In total, 34 species showed at least one case of melanism. The phylogenetic analysis indicated that the ancestral state of the family is non-melanistic, and that the melanic trait emerged independently several times, without being maintained stably over time. This suggests that melanism is an adaptation that evolves repeatedly, but can also be counter-selected. On an environmental level, the results show that the average daytime temperature is negatively correlated with the presence of melanism. The trait appears more frequently in cold environments. No significant correlation emerged from diurnal temperature range, precipitation, or latitude and longitude. But the data do not indicate a clear relationship between climate and the frequency of melanism in species that possess it, suggesting that other factors, such as life strategies or predation pressure, also come into play. Melanism may indeed provide a thermal advantage in cold environments, but it may present costs in terms of visibility to predators or physiology. Despite being associated with greater efficiency in thermoregulation, its presence remains rare and selective. This work extends the evidence of the adaptive role of melanism in reptiles, particularly in viperids, and confirms the importance of body colorations as complex responses to specific environmental conditions. We move to Brazil to discuss seabird strandings. These animals are often long-lived with life expectancies that can exceed 60 years, low fecundity, late sexual maturity, and colonial nesting. They live in dynamic environments and are subject to both natural and anthropogenic pressures on a local and global scale. On Brazilian beaches, strandings of these animals are a common phenomenon, but mass strandings with many individuals in a short time remain rare and complex to interpret. A group of researchers from the Federal University of São Carlos analyzed the spatiotemporal distribution of the abundance, richness, and diversity of seabirds stranded along over 1,000 kilometers of coastline between 2016 and 2019, assessing the potential environmental and anthropogenic factors involved. The data come from the Santos Basin Beach Monitoring Project, which operates daily. Live individuals and carcasses were considered, classified into 3 ecological groups: resident species, southern migrants, and northern migrants. Environmental variables analyzed included wave height, sea surface temperature, wind intensity and direction, and chlorophyll concentration, an indicator of primary productivity. As for anthropic variables, naval traffic was included. In the analyzed period, 11,700 individuals belonging to 25 species were recorded. Strandings were more frequent in the southern mesoregions and in spring, followed by winter with lower levels in summer and autumn. Resident species dominated in spring, while northern migrants were more present in the southern regions. Southern migrants, on the other hand, showed a peak in winter in the central areas. At the environmental level, higher wave height, stronger wind, and chlorophyll concentration were associated with an increase in strandings, while higher temperatures were associated with a slight decrease. However, the effect of the variables differed between groups. Residents showed a positive correlation with wind, southern migrants with colder waters, and northerners were influenced mainly by waves and productivity. A clear downward trend in strandings emerged along the south to north gradient in all ecological indices analyzed. Variations between ecological groups suggest that reproductive cycles and migratory periods, together with food availability, are key factors in the distribution and observed mortality. Human activities, such as ship traffic or the oil industry, did not show significant direct effect, but remain elements of vulnerability to be monitored. This is the first study to show which variables are linked to strandings, providing essential tools for the conservation of numerous marine species. We move north of the American continent to talk about the link between plants and pollinators. It is well known that the climate crisis is dramatically reducing pollinator populations. One of the most adopted solutions is to sow wildflowers in cities to offer new resources to insects. But a study by the Ohio State University, in collaboration with the University of Cambridge, highlights a little considered aspect. Not all wild plants are beneficial to pollinators, mostly if they grow on contaminated soil. The study was conducted in Cleveland, on abandoned lots that were once home to heavy industrial activities, such as steelmaking, refining, and car manufacturing. These soils, now poorly managed, host spontaneous vegetation that attracts bees and other pollinators. Many of these plants are hyperaccumulators, capable of absorbing heavy metals from the soil in roots, shoots, and leaves. However, it was unknown whether these metals could reach nectar, the main food source for pollinators. To find out, researchers sampled flowering plants from 8 neighborhoods and analyzed the nectar and sap in the laboratory using mass spectroscopy. The results were concerning because all the species examined were found to have metals in their nectar. The metals found most frequently and in the highest concentrations were lead, chromium, cadmium, and arsenic. Levels varied among plant species, but also within individual habitat patches, and metal concentrations could vary greatly, making it very difficult to predict the distribution of trace metals. The consequences of these findings are not negligible. Even low concentrations of metals can have long-term effects on pollinators. Cognitive impairment, learning problems, reduced flight ability, delayed egg laying, and high mortality in the clutch. Metals can accumulate in organisms throughout their life cycle, exacerbating toxic effects over time. Some metals also alter foraging preferences and can affect the entire ecological network between plants and insects. This doesn't mean that wildflowers should be avoided, but that soils must be analyzed before starting urban reforestation programs or planting areas for pollinators. If contaminated, they must be remediated to prevent apparently favorable habitats from turning into ecological traps. The study suggests a more integrated approach, in which soil health is considered an essential part of designing urban environments for biodiversity. We continue to explore the link between plants and insects, this time focusing on Lepidoptera. A new international research introduces the Salient aroma hypothesis, according to which the odors emitted by plants influence the food specialization of butterflies and moths. The idea starts from a simple fact. The quantity of odors and therefore of chemical information produced by plants and useful for insects for localizations changes, particularly between day and night. Consequently, insects must have evolved specialized structures to identify particular odors among others. To test this hypothesis, the researchers first confirmed through a meta-analysis of the literature that the emissions of volatile compounds by plants are more abundant and diversified during the day. Subsequently, they examined 94 species of Lepidoptera, analyzing the size of the antennae, key olfactory organs, of over 580 specimens and collecting information on their host plants. The results show that antennae vary in relation to sex, diurnal and nocturnal activity, and the degree of food specialization. In detail, females active during the day have larger antennae than those active during the night. This is consistent with the fact that plants release a greater quantity and variety of odors during the day. Females, which have to find the right place to lay their eggs, rely heavily on odors to identify the correct plants. If they are specialized, that is, tied to a few plant species, they must be able to accurately recognize the characteristic odor of their host plant, even when it is surrounded by many other signals. For this reason, having more developed antennae helps in chemical discrimination. Consistent with the research's hypothesis, specialist females show larger antennae than generalists, probably to refine this recognition ability. In males, on the contrary, an opposite trend is observed. Generalists have larger antennae. This could depend on the unpredictable distribution of females in species without a close association with the host plant, a situation that requires greater reliance on chemical signals to locate them. Daytime activity is also linked to diet. Diurnal species, immersed in an odor-rich environment, tend to have a more specialized diet, while nocturnal species, which encounter fewer chemical signals, are more generalist. The study highlights how the interaction between plants' emission and olfactory capabilities may have shaped the evolution of feeding strategies in Lepidoptera. Although there are exceptions, such as some strongly odorous nocturnal herbaceous plants and their specialized herbivores, the results suggest that the availability of plant odors represents a crucial factor in the evolution of ecological specialization. The Salient aroma hypothesis is therefore proposed as a new interpretative framework to understand how plants influence not only the behavior, but also the evolution of the insects that depend on them. This concludes today's episode of Drops of Science. Thanks for staying with us. See you soon, and good science!